Hello guys, and in this video we'll be going over the electron transport chain. This is the step after the citric acid cycle. So there's glycolysis, the citric acid cycle, and then there's the electron transport chain. Basically what's going to happen is you're going to make NADH and FADH2 from the citric acid cycle and glycolysis, and those electron carriers, FADH2 and NADH, are going to deliver their electrons to the electron transport chain, which is eventually going to lead to the production of ATP, which is your energy currency. So background here, you need to know what NAD and FAD are. Nicotinamide adenine dinucleotide and flavin adenine dinucleotide. The reason why they're called these is because you have a base, a sugar, and a phosphate, and there's two of them. And that's why it's called dinucleotide. Once again here, base, sugar, and phosphate, two of them, dinucleotide. All right, these are going to be either oxidized or reduced, and in their oxidized form, they're going to be written as NAD plus or FAD. In their reduced form, they're going to be written as NADH or FADH2. And if you don't know already, reduction is the gain of electrons, and oxidation is the loss of electrons. And where is this going to take place? In the mitochondria. The mitochondria is your energy producer. It's going to have two membranes. There's the outer membrane and the inner membrane, and then there's the matrix. Then basically the inner membrane is where the electron transport chain is going to take place. All right. So here we are. This is a zoomed in view, as in if we were to look at, we'll compare it to this. This is a zoomed in view of what's going on. And there's these various proteins that are embedded in the inner membrane. And those proteins are going to receive electrons from either NADH or FADH2. And they're going to basically transport those electrons down to the next complex. So there are different complexes. They're called complexes. So there's complex 1, complex 2, whatever. They're going to transport their electrons down the various complexes, and in the process of doing so, there's going to be protons that are going to be pumped from the matrix to the inner membrane space, and that gradient, electrochemical gradient, is going to create, basically is going to make the protons want to flow back into the matrix to balance out. And in doing so, they're going to generate ATP by causing this this turbine-like protein known as ATP-synthase to spin and add a phosphate to ADP to create ATP. All right. So let's see here. Succinate to fumarate. That's part of the citric acid cycle. That's where you're going to have FADH2 that's going to basically donate its electrons to complex 2. So it's a big distinguisher between NADH and FADH2 in that NADH donates its electrons at complex 1 versus FADH2, which donates its electrons at complex 2. All right. What does that tell you? Based on what we'll cover, you'll put 2 and 2 together and understand that one of them basically gives you more energy than the other. All right. So at complex 1, NADH passes its electrons right here, right? So it goes from NADH to NAD plus, meaning it gets oxidized or reduced. So let's see here, NADH to NAD plus a proton. Is that an oxidation or a reduction? Did it lose or gain electrons? If you said lose electrons, then you're correct. And in the process of doing so, a proton is pumped. So and then complex 1 is going to donate its electrons to something known as Q. Q, also referred to as Ubiquinone, is going to be a mobile carrier, and it's not going to pump protons. So these mobile carriers are simply going to take the electrons from one complex to the next, and then a proton is going to get pumped again. So each step is going to generate a proton pumping event, and eventually you're going to have the final pumping of a proton, and also the final electron transporting to oxygen. Oxygen is going to be the final electron acceptor, and it's going to generate water when it gets its electrons. So how do these electrons get transported down in one direction as opposed to another direction? The reason is something known as redox potentials. So each of these proteins, so 1, 2, and 3, have different redox potentials, meaning that they have different electron affinities. So complex 1 has has a lower affinity than complex 2 for electrons. That's why you basically go in one direction and not the other. So the redox potential differences are the reason why you go unidirectionally. So the way it goes is complex 1, electrons received, proton pumped, ubiquinone, also known as Q. Then you go here to complex 3 because complex 2 is another story. We'll talk about it in a second. Complex 3 pumps a proton, and then you go to cytochrome C, which carries the electrons to complex 4, also known as cytochrome C oxidase. Then you pump a proton, then the electrons are given to water, and you've generated a electrochemical gradient. Electro, in a sense that there's a difference in charge because you have plus charges that have accumulated in the inner membrane space, as opposed to the matrix, and chemical in the sense that there's a lower pH here because protons are acidic, and that gradient now is going to allow for these protons to flow through ATP-synthase to cause it to spin and attach a phosphate to ADP to cause a production of ATP. All right? So for FADH2, it's going to donate its electrons at complex 2 as opposed to NADH, which donates its electrons at complex 1. The reason why I said earlier that makes a big difference is because you notice that you don't pump a proton in that case here, right? You notice you only pump 2 protons here as opposed to, let's say, 3 here with NADH where it starts, all right? And so also remember that complex 2, this reaction here, is part of the citric acid cycle where you go from succinate to fumarate, all right? So to summarize once again, your electron carriers are going to donate their electrons from complex 1, let's say NADH does, complex 1 to Q, also known as ubiquinone, complex 2 to complex 3, protons are being pumped once again as a result of the electrons being transported to cytochrome C, which is also a mobile carrier like Q ubiquinone, to cytochrome C oxidase, also known as complex 4. Then the electrons eventually end up at water, at oxygen to make water, and then you have a gradient that's generated, and then those electrons flow through ATP synthase. ATP synthase spins and attaches inorganic phosphate to ADP to make ATP, and that's your energy currency of the cell. Thank you for listening. If you have any questions, please send them in, and don't forget to please subscribe.